Hi everyone, I'm Alex Marras. I'm a postdoc at University of Chicago, and I'm going to talk about polyelectrolyte complexation with nucleic acids. Um, and so we're interested in the phase separation of nucleic acids with the use of cationic polymers for tunable assemblies at the nanoscale and also at the microscale, both from a science of self-assembly standpoint and from a therapeutic standpoint. Um, Most of the work that I'm showing here today is using oligonucleotides or very short nucleic acids that are useful for controlling gene expression through things like antisense, oligos, and microRNA. And these could be very useful and very impactful for gene therapy, but they haven't been fully realized due to the major challenge of delivery. And so the biggest problems are avoiding nucleus degradation and biodistribution. Um, the two kind of gold standards right now are chemical modifications to the nucleic acid backbone that resists the nucleus degradation, but doesn't do anything for distribution, and hydrophobically driven assemblies that have proven useful in liver targeting and local injections. So there's been a lot of success with both of these, but they do have their limitations. And so the approach that we're using is polyelectrolytes, charge-driven assemblies of hydrophilic materials. I think this audience is plenty familiar with this, but these can protect our cargo and also improve the biodistribution through programmable size and morphology. And this is actually proven successful in cell studies, both in our lab and in other labs. But we don't fully understand the assembly process or how to control the physical features that are going to be important for distribution and delivery. So our goal here is to understand how the physical and chemical properties of each component will ultimately affect the assembly and stability of the particles. So the materials we're looking at for the cations, we're comparing polylysine and PBBTMA, this synthetic polycation. Some of the major differences between the two of these are that PBBTMA has a higher charge density and is more hydrophobic, and they use a different charge group. And also on the nucleic acid side, we're looking at some of these common therapeutic modifications that I mentioned earlier. One being, well, the difference between DNA and RNA first, is methoxy modification, and also this phosphorothioate modification for all of these. So what we found, early work shows that one thing that is affected is the morphology. So if in the case of single-stranded DNA, this is just the bulk system here, just polylysine and DNA. When DNA is single-stranded, you see these liquid-like coacervate droplets on the microscale. But when this is hybridized into double-stranded DNA, then you see these solid-like precipitates. So this phase is really controlled by the hybridization of the DNA. And then on the nanoscale, when you're using PEG and micelles, you see a similar thing where the shape of the micelle is controlled by DNA hybridization. So for both cations here, we see spherical micelles through TEM and small angle X-ray scattering. And with double-stranded DNA, we see these long worm-like cylinders, especially with polylysine, and some shorter cylinders of packed helices for the PBBTMA. And so when we look at the nucleic acid backbone and some of these common therapeutic chemical modifications, we see something pretty drastic where for, say, DNA and RNA, we see these spherical micelles, but when you add this 2' modification, it completely disrupts micellization, and you see these this kind of web of unimer complexes maybe. And a similar thing in the double-stranded case, even for double-stranded DNA, which we're starting to explore further. But then when you add that phosphorothioate modification, and again, this now enables that complexation, and you still get micelles, although they're not spherical anymore. So these small therapeutic changes actually have a pretty large impact on the assembly of micelles. So anyways, the third thing that we found is that the size can be controlled and is very dependent on some of the polymer lengths. So this is sort of a summary of all the core sizes from small angle X-ray scattering that we found for all these cases. And what we end up seeing is that the length of the DNA across the board, regardless of the polymer choice, the length of the DNA is independent on the size of the micelle. But this is actually highly dependent on the length of the cation, or what I think is actually just the charge block of the block polymer. I think this is what is going to control the size of that micelle. And maybe loosely the size of the peg or the neutral block. This is a whole other study that that I'm also doing, that I'm happy to talk about another time with anybody who's interested. But we're starting to really now develop some of these experimental scaling laws based on the length of each component in these systems. And then, in addition to the morphology and the size, we also see complex stability changing with the choice of polymer. So this is the bulk system, so there's no peg here. As we increase the length of either polymer or the DNA, we see a transition from no complexes to these liquid-like coacervates. And as we go into even longer polymers, in the double-stranded DNA case, we see solid precipitates. But these transitions happen with much shorter polymers in the lysine case versus the PBBTMA, to the point where we only see solid precipitates with lysine. And then when we look at the micelles, this is now with light scattering, and we look at the stability of these micelles, we see that they're more resistant to salt with polylysine compared to PBBTMA. This result, combined with the last one, tells us that the ion pairing is actually weaker with PBBTMA versus polylysine, despite the fact that it has that higher charge density and additional hydrophobicity. When we look at the nucleic acid backbone modifications, we see across the board a big jump in stability due to the phosphorothioate modification versus the natural backbone for all of these cases. Which is, both of these things are very interesting, especially this drastic change from the nucleic acid, because oftentimes these are the things that you're actually delivering, where you might design the system with something like DNA, and having an understanding of how these will affect the stability of the complexes, both to protect the cargo and keep a stable carrier, but also ultimately to release the cargo. Having this understanding is very important. So just to wrap up here, there's three big things that we found with these complexes. The first thing is the morphology. This is driven largely by hybridization of the DNA or the nucleic acid, and also is affected by the backbone chemistry of the nucleic acid. The size is largely driven by the charge block of the block polymer and independent of the length of the anion, or in this case, the nucleic acid. And the stability is very dependent on the molecular structure of the components you're using. So we found that PBBTMA was a little bit weaker than polylysine, and phosphorothioate made a much stronger ion pair with all the nucleic acids versus the natural backbone. So with that, I want to thank the Terrell Lab at University of Chicago and the funding sources and everybody that contributed to this work I showed today. And thank you very much for listening.